Okay, we'll make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we're starting a new unit and we're looking at cell membranes. Okay, and we're going to focus on cell membranes, osmosis, and facilitated diffusion. We're not going to be looking at any exam questions specifically in today's lesson, because it would only be worth looking at exam questions once we've covered active transport and cold transport as well, because that ties in very well with cell membranes. So we're looking at active transport and cold transport in our next lesson. And then if we have time as well, we'll have a look at some exam questions then. Today, we're just going to look at cell membranes, structure, everything about the cell membrane. We're going to look at osmosis and facilitated diffusion as well. Okay, this is the part of the spec that we are looking at. And as you can see, this part of the spec is literally just going over the structure of the cell membrane and what a cell membrane consists of. And you get common exam questions behind what the structure of the cell membrane looks like and why specific components are in place. You then need to know about the processes of movement across the cell membrane, and we're partly going to start looking at that in today's lesson. We're going to look at simple diffusion, facilitated diffusion, and osmosis. Next lesson, we're going to look at active transport and cold transport. Okay, and we will also cover this final part of the spec over here. Okay. So if we make a start, this is a cell membrane. This is an image of a cell membrane that we have over here. And there are many different components in this cell membrane that you need to be aware of. You need to know what glycoproteins and glycolipids are. You also need to know what the what a peripheral membrane protein is and an integral membrane protein is, or an intrinsic and an extrinsic protein. You need to know the function and purpose of cholesterol within the cell membrane. You also need to know what channel proteins are, and also the fluid mosaic model of this and the phospholipid bilayer. And that's what we're going to go through in today's lesson to start off with. Um, so again, this is specifically focused on AQA. However, there's a lot of overlap between the exam boards, so you would just have to decide if this is content that you will need to know by looking at your specification. Most of the content does overlap. Okay, so if we begin now. Okay, so we're going to start off with a key definition on the specification, and that is the fluid mosaic model of the cell membrane. Okay, now this cell membrane, okay, it can be described as being the fluid mosaic model. And what does the fluid mosaic model of the cell membrane actually mean? Well, the fluid mosaic, you need to look at them separately. What does fluid mean and what does mosaic mean? Okay, so plasma membranes or the cell membrane can be described as the fluid mosaic model. Fluid, what does that mean? You need to define it. Mosaic, what does that mean? You need to define it. And whenever exam questions are asked this, they will normally be worth 2 or 3 marks. And the way to gain maximum marks on questions like this is you need to define what the fluid is and what mosaic means. And then once you've defined them, that will get you your marks. So we're going to start off by defining what fluid means. Fluid means phospholipids are able to move relative to one another, giving the membrane a flexible shape. Okay, so the membrane, the plasma membrane, what we have in cells, the plasma membrane is actually made out of phospholipids. Okay, and remember, if we think back to biological molecules, we looked at fats, we looked at triglycerides, and we looked at phospholipids. Okay, we looked at them in biological molecules, and one of the uses that we saw phospholipids having is that they are a component of the cell membrane. They are the component that make up the plasma membrane or the cell membrane, and that's why we're looking at them now. Okay, so the plasma membrane is made out of phospholipids. And the reason why it's described as being fluid is because the phospholipids, which are lined against each other, we looked at a diagram then as well, but as you can see, can you see these are phospholipids? Each individual line is a phospholipid. Okay, And those phospholipids are lined next to each other and they're able to move. And because they're able to move relative to one another, the membrane can bend and have a flexible shape. So that's the purpose of the plasma membrane being fluid. The fluidity of the membrane allows it to move and bend, giving it a flexible shape. Okay, so that's the first definition. Phospholipids are able to move relative to one another, giving the membrane a flexible shape. That's why it's called fluid, um, in terms of the fluid mosaic model. Now, why is it called mosaic? Now, you've probably heard of mosaic in the in GCSE biology. Okay, and where you've heard of mosaic, I'll describe to you. You probably heard of tobacco mosaic virus, TMV, and that affects plants. The leaves start to develop a mosaic pattern on their surface. Okay, mosaic is just a pattern. Okay, it's just a type of pattern that that's what mosaic means. Okay, so why is it called the fluid mosaic model? Well, the fluid mosaic model, if we think about it, this is what it means. In the plasma membrane, you have many shaped sort of components. Okay, so as you can see, we have some integral membranes, we have some peripheral membranes, we have some cholesterol, we have some channel proteins, we have some glycoproteins, we have some glycolipids. We have many different components that are surrounding this plasma membrane. Because we have so many of these components, they vary in shape, size, and pattern. There are many different shapes that are embedded within this membrane. And because you've got that variety of shape, it starts to turn out like a pattern. Okay, and that's why it's called mosaic. Yeah, and if we combine them together, the fluid part and the mosaic part, combine them together, you get the fluid mosaic model. Okay, fluid means that the phospholipids which make up the plasma membranes, they are able to move relative to one another. And mosaic means you've got many different sized proteins which are embedded in the membrane. They will vary in shape, size, and pattern, and that means you have a pattern developing. Okay, and that's why, in summary, it's called the fluid mosaic model of the plasma membrane. Okay, this is a common exam question. Okay, you will get asked a question like this in your either paper one or paper two, in or paper three. Okay, because this is a very common exam question. You will find model mock scheme answers if you search this one up as well. And we will have a look at some of those exam questions in our next lesson. So that's the fluid mosaic model of the plasma membrane. It's a really important concept. What we're going to move on to now are the components of the cell membrane. So as you can see, if we look at this image, we can see that there's many components of the cell membrane. We're now going to move on to them in a little bit more detail and explore what each of those actually do. Okay, so the components of the cell membrane. We're going to start off with the most obvious one, and that is phospholipids, the one we looked at in biological molecules. Okay, phospholipids, they allow lipid-soluble substances to enter and leave the cell. They prevent water-soluble substances entering and leaving the cell. So if we have a look at this picture over here, we can see something that's called the phospholipid bilayer. And this is a really important phrase that we have. The phospholipid bilayer just describes all the phospholipids which are lined up next to each other. They form this structure called a bilayer. And that's all the phospholipid bilayer means. The phospholipids are lined up next to each other, forming a bilayer. That's the name of the structure. Okay, and phospholipids, if you think back to biological molecules, they have a structure that have two fatty acids, they have a phosphate group, and they have a glycerol. Okay, that's the structure of a phospholipid. And remember, the phosphate group is hydrophilic, which means it can interact with water. And these circles over here, they're basically representing the phosphate group. Okay, these are representing the phosphate group. that can interact with water. And the lines which are pointing inwards, they're representing the fatty acids. Okay, so if we sort of zoom in onto that structure, we have something like this. We can see something like this. And what that means is that these are your two fatty acid tails. And remember, fatty acids can only interact with fats, they can't interact with water. And that's why they're pointing inwards, because they can't interact with water. And the outside is the phosphate group. That circle is representing your phosphate group. Okay, and your phosphate group is charged. Because it is charged, it can interact with water. Okay, so phospholipids, they will allow lipid soluble substances to enter the cell. Okay, so because in the middle we've got a phospholipid bilayer that only has that can only tolerate fats, it can't allow water to enter. You can't allow water to enter because of that those fatty acids that are in the middle of the bilayer. Those fatty acids won't mix with water. Okay, only charged substances or really small substances can mix with water. Fatty acids, they will not allow water through. Okay, so only fats, only lipid soluble substances can enter and leave the cell because of that bilayer. Water will have to go through another pathway. Okay, water can't just simply just go across the cell membrane. It has to go through a specific pathway, and that's what we're going to look at later on in today's lesson. So we've looked at phospholipids. The next thing, which is really important, is cholesterol, and they like to ask what the function of cholesterol is in cell membranes, and it's a very much ignored factor that many people don't pay attention to. Cholesterol restricts the lateral movement of molecules in the membrane. Lateral means next to each other. So what does that mean? You want the membrane to keep its structure, right? You want the membrane to be stable, a bit flexible. However, you don't want it to sort of be so flexible that there's no purpose of having that cell membrane. If it's very flexible, then it will sort of break apart. You don't want it to be very flexible, and that's the purpose of cholesterol. The cholesterol will restrict the movement of molecules in the membrane. You don't want molecules to be moving around in the membrane. You want it to have that solid structure. You don't want channel proteins moving from place to place. You don't want intrinsic proteins to be moving. You don't want glycoproteins or glycolipids moving or anything like that. So the cholesterol will make sure that the movement between the molecules in the membrane is reduced. Okay, that means that they will become less fluid at high temperatures. This means that water and dissolved ions can't leak out of cells. What does that mean? If you increase the temperature, what can happen is that the membrane can melt. If we think about it in sort of basic terms, the membrane can melt. We don't want the membrane to melt. That's why the cholesterol will keep its rigidity in the membrane, providing it with its stiff structure and preventing it from losing its strength so nothing can sort of leak across the membranes. Okay, people who have sort of water retention problems will have leaking membranes. Okay, the membranes will be weak and the water will just leak across the membranes. Okay, so those are two important components of your cell membrane, the phospholipids and your cholesterol. The phospholipids are your lipid-soluble substances that will allow anything that is lipid-soluble to move across the membrane. It won't allow water to move across the membrane. Your cholesterol will restrict your lateral movement, ensuring that the membrane doesn't become leaky at high temperatures. Okay, now the next thing we're going to look at is peripheral proteins, and these are extrinsic proteins. And if we have a look at this diagram, the extrinsic proteins are these proteins over here. These proteins are your extrinsic proteins. And as you can see, they are not like going across the whole cell membrane. They're only forming a part of the cell membrane. Okay, they're only forming a small aspect of the cell membrane. They're not like going through all the way of the cell membrane. Okay, so that's the first factor. Extrinsic proteins are external. They don't go through the whole structure of the cell membrane. Okay, and the purpose of them is that they will provide mechanical support. Okay, so as you can see, think about alloys from GCSE chemistry. Alloys, the purpose of alloys is you have many different sized atoms that are present, and that will give the metal a stiff structure, preventing it from snapping or anything like that. It won't be any malleable, it won't be bendy or anything like that. And that's because you have different sized atoms. Here, these peripheral extrinsic proteins, they will provide mechanical support because they literally just come into the membrane and just stick in the membrane. Okay, they're not even going through the whole distance, they're just sticking in the membrane. Okay, they're like stones sticking in the membrane. And what they do is, as you can see, they're different sized. So that's why they provide the mechanical support. Just think about alloys and it will be very understandable. And these peripheral proteins, they will be connected to other proteins or lipids to make glycoproteins and glycolipids. Okay, so as you can see over here, this thing over here is a glycoprotein. This thing over here is a glycolipid. Basically, once you've got an extrinsic protein, once you've got a peripheral membrane protein or an extrinsic protein, that protein will be connected to another protein or it will be connected to a lipid. And the reason why it's connected to those forms like a lipid or a protein is the lipid and the protein will act as receptors. So anything which needs to activate the cell can bind to those lipids and proteins and can enter the cell. Okay, that's the purpose of glycoproteins and glycolipids. Glycoproteins and glycolipids, they're literally attached to your extrinsic protein and they act as receptors. So if anything needs to enter the cell, they will bind to the glycoprotein or glycolipid, they will act as receptors, and then they will enter the cell. Okay, that's the purpose of your glycoproteins and your glycolipids. Okay, so it's needed for cell recognition and so can act as receptors. So those are your peripheral proteins. Okay, now if you've looked at extrinsic proteins, there's another type of protein. We have intrinsic proteins. Okay, and intrinsic proteins are basically, if you look at this diagram, we know that the extrinsic proteins, they don't span the whole length of the cell membrane. They only span a little bit of the cell membrane and they provide that mechanical support to the cell membrane. The intrinsic proteins, they will be present on the inside of the plasma membrane. They will be present all the way through the plasma membrane from one side to the end. That's where they will be present. Okay, so that's where your intrinsic proteins will be present. So intrinsic proteins are protein carriers or channel proteins. So these things are known as channel proteins. Okay, and if you look at the image, channel proteins, they literally act as a channel to go through. Okay, they literally look like a channel and act as a channel as well. And their function is as well involved in the transport of molecules across the membrane. Okay, so molecules need to be transported across the membrane. So they will go, some will just literally simply diffuse across the cell membrane or the plasma membrane. Some will have to go through this channel protein. So let's think about water. Remember, I said that this is a phospholipid bilayer. Center, you have your fatty acids. The fatty acids can't tolerate water. They're not going to let water diffuse across the cell membrane. But the cell needs water. The cell can't live without water. Okay, it's a really big requirement for the cell. So how does water get in? Well, water goes through the channel protein. The channel protein will allow the water to move through from one end to the other and into the cell. Okay, because the phospholipid bilayer doesn't allow the water to move across, so what? The channel proteins are there, they will allow the water to move across. And that's what the channel proteins are there to do. Anything which can't diffuse across the phospholipid bilayer, the channel proteins will come in and then they will transport that if it's reasonable and it is required by the cell. And there are two types of channel proteins. Okay, you have your protein channels, and you have carrier proteins. Okay, these things I've labeled as channel proteins, they're an example of protein channels. Okay, if we look at the inside, these gaps in the inside, they will be more likely to be carrier proteins. So channel proteins, you have two types. You have protein channels. These are tubes which fill with water to enable water-soluble ions to diffuse across. So these tubes that we have over here, they will fill up with water. Once they've filled up with water, the water can allow anything which is water soluble, such as mineral ions, to diffuse across and enter the cell. Okay, and whenever there's water required, water can just go through this channel protein. Okay, so that's protein channels. You also have carrier proteins. Okay, and this we will come across again next lesson when we look at co-transport. Okay, because it's a big aspect of co-transport. But basically, carrier proteins, they bind with ions which are not water soluble or larger molecules such as glucose and amino acids. These will then change shape to transport them to the other side of the membrane. Don't worry about carrier proteins at the moment. We're going to look at them in more detail in our next lesson. But essentially, carrier proteins, they are activated by an ion. An ion will bind to the carrier protein, and if the ion binds to the carrier protein, what will happen is that the carrier protein is activated, it can change shape, and it can release something onto the other side. For example, glucose amino acids, they're very big. They can't just simply diffuse across the cell membrane because they're really big. So they will go to a carrier protein. They will bind to the carrier protein first. An ion will activate that carrier protein, and then the carrier protein will change shape, and it will take the glucose or the amino acid and it will put it onto the other side. That's how carrier proteins work, but we will look at that again in our next lesson. So those are basically the components of the cell membrane. Okay, so we've looked at a lot there. We've looked at the fluid mosaic model. We've looked at the phospholipids, the cholesterol, the extrinsic and intrinsic proteins, the glycolipids and the glycoproteins, and we've also looked at channel proteins. Now what we're going to move on to is a bit more on the partially permeable membrane, and then we have a bit to cover on simple diffusion, facilitated diffusion, and osmosis, and then we will call it a day there. So partially permeable membrane, okay? So we know from GCSE that exchange across a membrane, osmosis, for example, diffusion occurs across a partially permeable membrane. Now, across the membrane, we know that there are phospholipids, okay? Now, the phospholipids will allow lipid-soluble substances to diffuse across. And that's basically what this is saying. The plasma membrane is made up of a partially permeable membrane. And what that actually means is that phospholipids are on the plasma membrane. And the phospholipids are the things which make it partially permeable because they can only allow lipid-soluble substances to diffuse across. Okay, so an example of a lipid-soluble substances is hormones or very small molecules such as oxygen and carbon dioxide. These are lipid-soluble and they can just simply diffuse across the plasma membrane. Things like ions, okay, potassium ions, sodium ions, these ions, they're not lipid-soluble, okay, because they are charged. Anything charged is not lipid-soluble. Because these things are not charged, they will have to go through the channel protein. They will have to be dissolved in water so they can be transported to the other end, or they will have to go through a carrier protein to get transported into the cell. Okay, so water-soluble substances, which are polar, they can't just simply diffuse across the membrane. And large molecules such as glucose or amino acids, they cannot pass through as well by simple diffusion. Okay, because remember that phospholipid bilayer is only going to allow substances which are lipid soluble to move across. It has to be small and lipid soluble. If it's not small and lipid soluble, for example, if it's something like water or if it's charged or if it's big, it can't go through. It'll have to go through a channel protein or a carrier protein in order to get into the cell. Okay, now what we're going to do is we're going to look at some simple diffusion. Now, simple diffusion is your GCSE diffusion. Okay, so definition is the exact same. It's the net movement of molecules from an area of high concentration to an area of lower concentration until equilibrium is reached. No ATP is required for this to happen. Simple diffusion is literally just high to low concentration until you've got the same amount on each side. In that case, you won't have any net movement. Okay, and it's a passive process, which means no ATP, no energy is required for this process to occur. Simple diffusion only occurs with fluids. The reason why it can only occur with fluids, gases and liquids is because fluids have kinetic energy. That means the particles are able to move. It can't happen with solids because solids, the particles in solids can't move. They're literally fixed in one position, so they can't move from one place to the other. In fluids, they can move from one place to the other because they're free moving, they have kinetic energy, so they can be transported across the membrane. The molecules must be small and lipid soluble. So remember when I've been saying simple diffusion across the phospholipid bilayer, that just means that across the phospholipid bilayer, only lipid soluble and small substances can simply diffuse. The reason why you need to have the simple before diffusion is because there's another type of diffusion, and that's called facilitated diffusion, which we're going to come on to in the next slide. So simple diffusion is just your GCSE diffusion. Facilitated diffusion is also a passive process. No ATP is required. It's basically like diffusion, but no ATP is required. However, when we say simple diffusion, that means substances can just diffuse from one side of the membrane to the other side of the membrane. Done. That's it. Okay, that's what happens. In facilitated diffusion, proteins are used to transport the molecules. Okay, so it could be a carrier protein or a channel protein or something like that. A protein will be used to transport molecules from one side to the other. Okay, so it's the movement of ions and polar molecules. Polar just means charged, which cannot simply diffuse, can be transported by membranes using facilitated diffusion. So you normally have your protein channels and your carrier proteins. Okay, so if we have a look at this image over here, we've got facilitated diffusion taking place because we have carrier proteins. Okay, as you can see, we have carrier proteins and we have protein channels. Okay, and anything which is not lipid soluble or small will be transported using facilitated diffusion. Facilitated diffusion is when you have a high concentration moving to a low concentration. It's still the same concept. However, the process is occurring through a channel protein or a carrier protein or some form of a protein. Okay, that's the only difference with facilitated diffusion. Proteins are used to transport molecules, whereas in simple diffusion, they literally just move by themselves across the plasma membrane. Okay, and it will normally be of water, of ions, of polar molecules, things that are not lipid soluble. Okay, and finally, what we're going to look at in today's lesson is osmosis. Now, osmosis is basically the same as GCSE. Okay, but there is a new term that we are using, and that is water potential. So if we start off to look at osmosis, this is the final thing we're looking at in today's lesson. Osmosis is the movement of water from an area of high water potential to an area of low water potential across a partially permeable membrane. So the definition is the same, as you can see. However, there is just one key difference, and that key difference is terminology. You can't say high water concentration anymore. You have to say water potential. Water potential is the term used for a high concentration of water. Okay, and that's what's changed from GCSE to A level. They've just made the definitions a bit more fancy. So osmosis is the movement of water from an area of high water potential to an area of low water potential through a partially permeable membrane. So that part stays the same. You just have to replace the concentration with potential. So high water potential to low water potential. Okay, and water potential is just created by the pressure of the water molecules. Okay, so water molecules have a specific pressure that they sustain in the body. And the pressure of water molecules will be measured in kilopascals. Okay, pure water has a water potential of 0. Okay, the more negative the water potential is, the more solutes that are dissolved in it. Okay, so the higher the water potential, the purer the water. The more negative the water potential, the more solutes that are dissolved in it. And this is a really important fact because sometimes you could get some exam questions that basically state the you might be given the kilopascals of different solutions and you might be asked to work out where osmosis occurs. Okay, so you might be given one which has a negative 100 kilopascal water potential and one which has a water potential of negative 50 kilopascals. What does that tell you? The negative 100 kilopascals tells you that there are more dissolved solutes in it, which means that it has a lower water potential. And the negative 50 kilopascal solution tells you that there are less dissolved solutes present within the water. That means it has a higher water potential. So osmosis is going to happen from the negative 50 kilopascal solution to the negative 100 kilopascals solution. Okay, that's the direction that osmosis is going to occur. And that's what most likely you'll be asked. Which direction does water move? Where does the water potential occur? And it's basically an application to the question. Okay,